Well, look, welcome to this week's quick take, week in, whatever you like to call it, in clinical immunobiology, where I like to look at what's happening around the world of internal medicine, pick out the bits which are of immunological interest and use them to illustrate some fundamentals of immunobiology. So this week's paper that came out, pretty intense. I don't think anyone is really going to want to read it in detail unless you're an immunologist, but it does bring out some principles of immunodeficiency which I thought we could use to revise what is in my lectures and in fact in the upcoming book. So let's just remind ourselves of the principles of what happens if your immune defenses are down, and we divide this into four main pillars, if you like, of immunocompetence, namely that we need complement, we need phagocytes, we need antibodies, we need T cells, all of which allow us to fight various types of infections, and if one of those arms is missing, then you're susceptible to various types of pathogens. So if your complement system is down, particularly the alternate pathway, you're susceptible to Neisseria, classical pathway and common pathway, which is C3, bacterial, and classical pathway, immune complex disease, which is not really immunodeficiency, but I just put it in there to remind you. If your phagocytes aren't working, your neutrophils, your macrophages, then this is fairly nasty. You get high-grade bacterial and opportunistic pathogens, nasty fungal infections in places where they shouldn't be, like, liver and tissues and so forth. If your antibodies are deficient, this is the most common immune deficiency, which I'm sure everyone knows how to recognize, at least I hope so. Recurrent cynopulmonary infections with ordinary everyday pathogens like strep and staph and Haemophilus, etc. And you also get acute and chronic diarrhea due to, Campylobacter and Giardia and so forth. If your T cells are lacking, think AIDS type infections if you're adult, think SCID if you're pediatric. So the things we see are, if their T cells are deficient, so probably the worst deficiency to have, although phagocytes is pretty bad, fungi, viruses, protozoa, listeria and mycobacteria. And we're going to talk about TH subsets in a moment. So just to emphasize that Th17 is responsible for fungal defense at the mucosal surfaces, and Th1 cells more or less the intracellular type pathogens, and this is important for this particular paper. So this is the paper. It's a very interesting life's work for the senior author, where there is a particular interest in autoantibodies mediating immune deficiency diseases. So if you've been to the course, you may remember that the most obvious example of this, which is beautifully illustrating the role of Th17 cells, is the anti-cytokine antibodies to interleukin 17 and 22. So we'll talk about a bit about those in a moment. But this is a new immune deficiency driven by antibodies to interleukin 23, an autoimmune disorder causing an immune deficiency. So just to summarize this, these types of adult onset T cell type immunodeficiencies are really rare. I may have seen one or two in a lifetime as a clinical immunologist. So it's not something that you're going to go out and diagnose next week, but at least you could be aware of these particular immunodeficiencies and be able to divide it into basic, which arm of the immune system to take further. So this paper describes a cohort of patients with recurrent infections of the T cell type. So we've just revised what that is: mycobacterial, nasty bacterial, fungal, viral, pneumocystis, cryptococcus, a whole range of T cell type significant infections. And to cut a lot of research to a single sentence, anti-IL-23 antibodies seem to be responsible, and the stronger the inhibition of interleukin-23, the worse the type of infections were noted. Now, this has a little bit of a conundrum and I don't want you to get too scared at this point about the detail, but I have to mention that interleukin-12 and interleukin-23 share a subunit called P40, and interleukin-12, the P40 links to P35, and interleukin-23, the P40 links to P19. And so when you have antibodies to IL-23, they could potentially also block interleukin-12 and mediate their effects in that way, or they could just be directed to the P19 and have nothing to do with interleukin-12. But anyway, the authors of this paper had a look at that and decided or worked out that interleukin-12 blockade didn't really matter. It was the interleukin-23 that correlated best with immunodeficiency. Excuse me. All right, so that's the paper. You'll never see a patient, or very unlikely to see a patient. So what is in that for you, for the average internal medicine practitioner or trainee? Well, I think you need to know what interleukin-23 does, and I'll tell you why in a minute. I think you need to be aware of anti-cytokine antibodies and how they might cause disease, and how, in general, what diseases may contribute to autoimmunity, particularly autoimmune cytokine diseases. So the first question I think is the most important, and that is, what does IL-23 do? Now, in the course, I emphasize the important cytokines involved in T-cell differentiation. So you'll remember that undifferentiated T-cells will differentiate into Th1, Th2, Th17, T follicular helper, or indeed T regulatory T cells. We'll leave these two for the moment. And they do so based on the type of cytokines which are present at the time they are activated. So if interleukin-12 is present, they go to Th1 cells, which largely protects from bacterial and viruses, intracellular type infections, and that's relevant to today's talk. They can go to Th17 cells to protect from fungal infections, and they do so under direction of TGF-beta and interleukin-1 or interleukin-6. And you will see here comes interleukin-23. And although it doesn't promote Th17 differentiation, is very important for maintaining and sustaining Th17 cells. So if you block interleukin-23, you tend to inhibit the function of Th17 cells. We'll leave Th2 cells for the moment. Let's concentrate on Th1, Th17. Th2, mostly helminth and allergic disease, which doesn't concern us in this particular lecture. So at first sight, you'd say, well, if a patient has autoantibodies to interleukin-23, then they should get much the same sort of stuff as if you're deficient in Th17 cells. Or Th17 products, and we know from a condition called APOSED that you can get antibodies to interleukin-17 and 23, which predispose to this condition, chronic mucocutaneous candidiasis. But clearly these patients are getting a lot more than that, and this suggests that there is some Th1 inhibition going on, so maybe interleukin-23 is doing a little bit more than we have assumed. Maybe it's also important in the activation of Th1 cells. Well, this is getting into too much detail, so I don't want you to get bogged down with that, but it's a very interesting concept, and this is fairly new in biology. So I think I've just mentioned that. So why do you need to know about interleukin 23? I've emphasized it again in this year's lectures more so than previously because there are antibodies or therapeutics coming out which target interleukin 23 for managing diseases which are Th17 driven. So guselkumab and risankizumab are monoclonals which are licensed in Australia for the treatment of psoriasis. I'm sure they're licensed in other countries, no matter, depending on where you're listening to this. And so we need to know what interleukin 23 does for that reason. So we know that Th17 cells are important in psoriasis. And so antibodies, therapeutic antibodies to interleukin 23 can help in that condition. But this paper would suggest that if we block interleukin 23, we need to be aware of the possibility that these more nasty opportunistic pathogens may well come to the fore. So it's worth, if you're treating patients with an interleukin 23 blocker, being aware of this paper and looking out for the potential that there may be the patient may become susceptible to these opportunistic infections. Now, the second point is to know about anti-cytokine antibodies. I don't think you need to know a lot because we can't measure them outside of a research setting. You can't write a routine pathology request form and say anti-cytokine antibodies. They'll ring you up and say what you're talking about. Nobody does that, and nobody does do it. But it does illustrate some important aspects of immune biology. So in the course we talk about type 1 interferon, important in that initial innate protection from viral infection, and it has been found that anti-cytokine antibodies directed to type 1 interferons are part of the story of patients who have severe SARS-CoV-2, severe coronavirus disease, and there are some genetic deficiencies of that pathway which also predispose to bad SARS-CoV-2 infection. We've mentioned several times interleukin 17 and interleukin 22 as being responsible for CMC. These other ones are a bit more fine print, but I put them there for completeness. But if you're aware of the first two, I'll be very happy. So which conditions are associated with anti-cytokine antibodies? There are two main ones, and they're also associated with other autoimmune diseases too. So it's not just cytokine antibodies. There are lots of multiple autoimmune diseases associated with APOSED and thymoma. Now you have to be aware, I'm sure you do know, that thymoma and myasthenia gravis go together as an autoantibody-mediated disease. There's something about thymoma that promotes autoantibody generation. I haven't seen any clear explanation about why that is, but it is. So thymoma, and the other condition that I talk about in the course is APOSED or APS1 or air deficiency. And if you remember the talk on T cell selection, air is responsible for turning on tissue-specific antigens in the thymus which allow autoreactive T cells to be deleted against various tissue-specific antigens throughout the body. So without air, you don't get that deletion. You get the release of potentially autoreactive T cells, and in this particular circumstance, anti-cytokine antibodies can be generated, as can antibodies to other endocrine organs like the parathyroid and the adrenal and so forth. Okay, so that's all I want to say about this particular paper. So just to summarize the important revision points for general medical trainees or practitioners, just revise the cytokines relevant to T helper cell differentiation. Today we're talking particularly about the role of interleukin 23 and Th17 cells, but be aware of those other cytokine influences because they all have clinical and therapeutic import. Note the relationship between Th17 deficiency and Candida. We talk a lot about that in the course. The conditions that predispose to autoantibodies, be aware of thymoma and APOSED or APS1 or air deficiency. And the new point, I guess, which would be, fine print, I suppose, is that IL-23 looks like being more important than just Th17 cell biology. It looks like it's going to have a role in Th1 cell development, and so those patients who are on anti-interleukin 23 just need to be aware that this is a possibility. All right, well, I hope you enjoyed that revision of immunology. If you haven't been to the course, then please put this in your diary. We go live on September the 14th, and all the videos will be accessible at that point. All information you can get off our website. These are the lectures that will be today's revision lecture it was drawn from, and so we'll re-advise all those concepts on those 3 lectures, and sometime in the future the same concepts will come out in book form for you to read. So look out for that in the future. So that's all I've got to say for this week. I hope you found that useful, and I'll be back with another mini lecture very shortly.